If the body had a set of construction toys, they would be neurons and synapses. Just like a child builds and destroys towers and structures, the body makes a neural circuit, tears it apart, and builds another one. Neurons are the primary parts, and synapses are the connectors. But these connections aren't solid. They are electrical or chemical. The connections occur in openings between neurons. It's hard to believe that little chasms are so important, but without synaptic gaps, nothing in your body would work. Neurons carry signals to muscles or organs, but they're not directly attached. There's an opening, a synapse, between the neuron and its target. We are not hardwired. We operate as a network of interconnected parts. Your body has approximately 500 trillion synapses. They come in two designs. Synapses are either chemical or electrical. Chemical synapses are much more common. In a chemical synapse, the break between neurons allows the chemical release by one neuron to temporarily connect to another neuron and then disconnect. Chemical synapses are axon dendrite connections. They involve the release of a chemical, neurotransmitter, which binds to a receptor. A lock-key matching of receptor and neurotransmitter allows many neurons to be in close proximity, but not all fire at once. The first neuron, this neuron is presynaptic. It's the one who is releasing the neurotransmitter. Let's call it Prentice, the presynaptic neuron. When Prentice releases a chemical, it's pushed out of the neuron, a process called exocytosis, and enters the extracellular space between neurons. The second thing that happens is the neurotransmitter diffuses across the synapse, and then third, binds with the receptor of the postsynaptic neuron. The postsynaptic neuron is the one post after the synapse. Fourth, the synapse, that is the space between the two neurons, is cleaned up through reuptake and inactivation. Reuptake means that some of the neurotransmitter is sucked back into the first neuron space, into the presynaptic neuron's axon terminals. Inactivation means that a small amount of enzymes are released to eat up the remaining neurotransmitter in the synapse. Problems with overbinding, staying too long, where the neurotransmitter stays attached to the receptor too long. And another problem, called overactivation, the reconnecting too often, called excitotoxicity. Both of these can damage or kill the neuron. Neurons can also get too close. When neurons get 10 times closer than a synapse, they trigger each other electrically. These electrical synapses are called gap junctions. These gap junctions are primarily axon to soma, axon to axon, or dendrite to dendrite connections. They can be found in several locations, including the retina and the cerebellum. The axon to soma synapses are often inhibitory. They make the postsynaptic neuron less likely to fire. To avoid gap junctions, the body insulates axons with a substance called myelin. Myelin is a fatty substance that looks white, as in white matter. Unmyelinated neurons look gray, as in gray matter. In the brain and spinal cord, glial cells, called oligodendrocytes, are glob-like dots of myelin that axons run through. It looks like silly putty with lots of straws going through it but not touching each other. Outside of the brain and spinal cord, myelinating SCHWANN cells wrap around an axon, leaving little gaps between them. It looks like an arm with lots of bracelets or armbands that don't touch each other. The gaps are called nodes of Ranvier.